Hello everyone. Welcome back to the Cell Culture series. In episode 13, we're diving into the fascinating world of 3D cell culture and organoids. Let's explore how scientists are creating more realistic models that better mimic the human body and revolutionize biomedical research. Let's get started. A 2D cell growing flat on plastic is about as realistic as a paper cutout of a human organ. But what if you could grow cells in 3 dimensions, forming structures that self-organize, develop gradients, and behave like real organs? That is exactly what 3D culture and organoids do. They are transforming drug development, cancer research, and personalized medicine right now. Flat cells, flat answers. The 2D problem. When a cell grows flat on plastic, its top surface faces media and its bottom surface faces plastic. In your body, that same cell would be surrounded on all sides by neighbors, blood vessels, extracellular matrix, and chemical gradients. The cell in your body is living in a rich 3-dimensional neighborhood. The cell on your plastic dish is living on a tile floor, alone, flat, and thoroughly confused about what it should be doing. The 85% problem. Up to 85% of cancer drugs that work in 2D cell culture fail when tested in animals. A significant part of this failure is attributed to the fundamental unrealism of 2D models, drug penetration, oxygen gradients, cell-cell signaling, and matrix interactions in a monolayer bear little resemblance to a solid tumor. 3D models are a direct response to this translational gap. Now, let's check your understanding with another question. Here's the question. What specific biological properties does a 2D monolayer fail to reproduce that a 3D spheroid can? A. 2D models cannot use human cells. B. Oxygen and nutrient gradients, cell polarity, extracellular matrix interactions, and drug penetration barriers that exist in real tumors. Circa 2D models cannot be used with cancer cells. D. 2D models cannot measure cell viability. The correct answer is B. Here's why. In a real solid tumor, cells at the center experience low oxygen, limited nutrients, and a completely different metabolic environment compared to cells at the outer edge. These natural gradients influence drug penetration, cell growth, and gene expression. In contrast, a 2D monolayer has no inner core. Every cell is exposed to nearly the same amount of oxygen, nutrients, and drug, making it much less representative of what happens inside the human body. The key takeaway: 3D spheroids recreate the complex environment of real tumors, making them a more reliable model for studying drug response and helping explain why many treatments that succeed in 2D cultures fail during animal or clinical studies. 3 levels of 3D complexity. Let's now explore the 3 major levels of complexity in 3D cell culture models. At the most basic level are multicellular spheroids. These are formed using ultra-low attachment plates or the hanging drop method. They usually contain a single cell type and are widely used to study tumor biology, drug penetration, and radiation response. The next level is organoids. These are created when stem cells self-organize within matrigel to form miniature organ-like structures. Because they contain multiple cell types and closely resemble real tissues, organoids are ideal for disease modeling, patient-specific drug testing, and developmental biology. The most advanced model is the organ on a chip. These microfluidic devices combine living cells with controlled fluid flow and mechanical forces, closely mimicking the physiological environment of human organs. They are extensively used for drug toxicity testing, pharmacokinetics, and ADME studies. In simple terms, spheroids model basic 3-dimensional tumors, organoids recreate miniature organs, and organ-on-a-chip systems simulate how real human organs function inside the body. The more complex the model, the closer it is to human physiology. Spheroids. Simple 3D. Multicellular spheroids form spontaneously when cells are denied a flat surface to attach to. Ultra-low attachment, ULA, plates have a hydrophilic coating that prevents cell surface contact. Cells aggregate and form tight spheroids within 24 to 72 hours. The result? A miniature tumor-like structure with a proliferating outer layer, a hypoxic middle ring, and a necrotic core, exactly mirroring what happens in a poorly vascularized tumor. The hanging drop method, an alternative spheroid formation approach. Place a small droplet, 20 to 30 microliters, of cell suspension on the inside of a plate lid, then invert the lid. Cells settle to the bottom of the hanging drop and aggregate by gravity into a single spheroid. No special plates needed, excellent for forming uniform spheroids of defined size. Organoids. Organs in a dish. Organoids are derived from stem cells or primary tissue fragments that are suspended in matrigel, a basement membrane extract from mouse sarcoma that gels at 37 degrees Celsius and mimics extracellular matrix. In the right hormone and growth factor conditions, stem cells self-organize into miniature organ structures with the correct cell type composition and spatial organization. Now let's take a look at some of the most widely used types of organoids and where they are applied in research. First, we have intestinal organoids. These are derived from intestinal stem cells and develop structures similar to the crypts and villi of the intestine. They contain specialized cells such as enterocytes, Paneth cells, and goblet cells, making them valuable for studying inflammatory bowel disease, microbiome interactions, and colorectal cancer drug testing. Next are brain organoids, generated from pluripotent stem cells. They form cortical layers containing neurons and astrocytes, providing powerful models for studying microcephaly, Zika virus infection, neurodegenerative diseases, and human brain development. Liver organoids are derived from liver ductal cells or hepatocytes and form bile duct-like structures. They are widely used for drug toxicity testing, liver disease research, and studies of metabolic disorders. Moving on to lung organoids, these are created from lung progenitor cells and develop alveolar or airway-like structures. They have become important tools for studying COVID-19, lung cancer, and cystic fibrosis. Finally, we have kidney organoids, which are also derived from pluripotent stem cells. They develop structures resembling kidney tubules and glomeruli, making them useful for drug nephrotoxicity testing and research into polycystic kidney disease. The key takeaway is that each organoid is designed to mimic a specific human organ, allowing researchers to study diseases and evaluate therapies in models that are much closer to real human tissues than traditional 2D cell cultures. Patient-derived organoids, personalized medicine in action. The most exciting application: take a tumor biopsy from a cancer patient, establish organoids in a dish, and test 20 different drug combinations on the organoids to find which kills the tumor most effectively. This approach, already in clinical trials, could match patients with effective therapies in weeks rather than the trial and error of current oncology practice. Now, let's test your understanding with another question. Here's the question: why is matrigel used as the matrix for organoid culture rather than tissue culture, plastic, or standard agar? A. Matrigel provides nutrients that cells cannot get from standard media. B. Matrigel is a basement membrane extract that gels at 37 degrees Celsius, providing the structural and biochemical cues that stem cells need to self-organize into organ-like structures. C. Matrigel is transparent, allowing better imaging. D. Matrigel prevents bacterial contamination. The correct answer is B. Here's why. Matrigel is rich in important extracellular matrix components such as laminin, collagen type 4, entactin, and essential growth factors. These molecules provide both the physical scaffold and the biochemical signals that guide stem cells to organize into complex, three-dimensional tissues. Another unique property of matrigel is that it remains liquid when kept below 4 degrees Celsius but rapidly forms a gel at 37 degrees Celsius, surrounding the cells in an environment that closely resembles the body's natural extracellular matrix. The key takeaway: unlike tissue culture plastic, matrigel doesn't just support cells, it provides the structural and biochemical environment that allows stem cells to develop into realistic organ-like structures. That's why it is the gold standard matrix for organoid culture. From seeding to 3D, building and testing spheroids. Spheroid formation is simpler than organoids and a great entry point to 3D culture. Here is the complete workflow from seeding to drug treatment. Spheroid formation protocol, ULA method. 1. Prepare ultra-low attachment round bottom 96 well plates. These have been coated to prevent cell attachment. 2. Seed 1,000 to 5,000 cells per well in 100 to 200 microliters complete media. Too few cells. Spheroids form too slowly or not at all. Too many. Spheroids fuse into irregular aggregates. 3. Centrifuge the plate at 300 grams for 3 to 5 minutes immediately after seeding. This brings cells to the bottom of each well and initiates aggregation. 4. Incubate 24 to 72 hours without disturbance. Cells aggregate and compact into a spheroid. Check daily by bright field microscopy. 5. Spheroids are ready when compact, round, with smooth edges. Diameter typically 200 to 500 micrometers after 3 to 5 days of growth. Drug penetration assay. Add fluorescent drug, or fluorescently labeled control molecule, at day 5. Incubate 24 hours. Section spheroid and image by confocal microscopy. Small molecules penetrate to the center, green fluorescence throughout. Large molecules or molecules with poor penetration properties accumulate at the rim only. This directly predicts whether a drug can reach hypoxic tumor cells in vivo. 4 mistakes in COVID lung organoids. Mistake 1. Using regular plates instead of ultra-low attachment. The fix. Ultra-low attachment, ULA, plates have a hydrophilic coating that prevents protein adsorption and cell attachment. Regular tissue culture plates have a hydrophilic surface that promotes attachment. Cells will simply form a monolayer instead of a spheroid. ULA plates are essential and non-substitutable for most spheroid protocols. Mistake 2. Disturbing matrigel before polymerization. The fix. Matrigel must be handled on ice at all times before use. Pre-cool all pipettes, plates, and tubes. Work quickly and transfer to 37 degrees Celsius immediately after plating. Moving, vibrating, or pipetting matrigel after it begins to gel produces a disrupted, uneven matrix that prevents proper organoid formation. Mistake 3. Growing organoids too large without splitting. The fix. Organoids develop a necrotic core when they exceed approximately 500 to 700 micrometers in diameter, as nutrient and oxygen diffusion limits are reached. Split organoids mechanically every 7 to 14 days by pipetting to break them up and replating in fresh matrigel. Treat organoid splitting as the equivalent of passaging cells. Mistake 4. Not validating cell type composition. The fix. Just because a structure forms in matrigel does not mean it is the organoid you intended. Validate composition by immunostaining for cell type specific markers, enterocytes, vilin, E-cadherin, Paneth cells, lysozyme, goblet cells, MUC2, for intestinal organoids. Without validation, you are measuring an undefined cell aggregate, not a functional organoid. During the COVID-19 pandemic, research teams used human lung and intestinal organoids to study how SARS-CoV-2 infects cells, identifying a CE2 receptor entry, testing antivirals, and characterizing cytokine responses, all without patient exposure. Patient-derived colorectal cancer organoids can now predict individual responses to chemotherapy with greater than 80% accuracy before treatment begins. This is personalized medicine becoming real. Summary. What you now know. 1. 2D culture is convenient but limited, 3D is more physiologically accurate. Flat monolayers lack oxygen, nutrient gradients, cell polarity, matrix interactions, and drug penetration barriers. Up to 85% of drugs that work in 2D fail in animals, partly because 2D does not capture tumor biology. 3D spheroids and organoids bridge this gap. 2. Spheroids are simple 3D, organoids are self-organizing, organ-like 3D structures. Spheroids form by simple cell aggregation, good for drug penetration in tumor models. Organoids require stem cells, matrigel, and specific growth factor conditions. They self-organize into mini-organs with correct cell type composition. Both are valuable. Choose based on the biological question. 3. Patient-derived organoids are enabling personalized cancer medicine now. Biopsies to organoids to drug testing in 2 to 3 weeks, identifying effective therapies for individual patients before treatment begins. Already in clinical trials for colorectal, pancreatic, and other cancers. This is the direct clinical application of everything in this episode. Thank you for watching the episode 13 of Cell Culture series. In the next episode, we'll dive deeper into drug screening, co-culture and advanced applications. See you there. If you found this episode useful, don't forget to subscribe our channel. Don't forget to like, share, and leave the comment below. We'll see you in the next episode. Thank you for watching.